Good morning, good afternoon, and good evening to alumni, parents, and friends from around the world. My name is Robin Gibbs. I'm a Regional Director of International Advancement at Brown and an alumna myself from the class of 1993. I hope that you and your loved ones are healthy and well. It's my pleasure to welcome you and to thank you for joining us for the second iteration of our International Advancement webcast series, The Race to Find a Cure for Malaria, featuring Dr. Jake Kurtis. We are so grateful to be able to continue to bring everyone together in these challenging times in support of the university and our students. With that, it's my pleasure to introduce our speaker, Dr. Jake Kurtis, who is an expert in infectious disease, and while on the precipice of finding a vaccine for malaria, has also answered the call to action in the fight against COVID-19. Dr. Jonathan Jake Kurtis is the chair of the Department of Pathology and Laboratory Medicine, director of the Warren Alpert Physician Scientist and MD PhD Training Program, and professor of pathology and laboratory medicine at the Warren Alpert Medical School at Brown University. Dr. Kurtis is a triple alumnus of Brown with an undergraduate degree in geology biology and a member of the first PLEMI class from 1989. Dr. Kurtis then earned his PhD in molecular parasitology in 1995 and his MD in 1996. And it was during this time that Dr. Kurtis traveled to Africa to do research with a Brown professor and himself contracted malaria. This, as we will learn more about, has informed his life's work. Subsequently, Dr. Kurtis completed his postdoctoral training in malaria immunology epidemiology at the Walter Reed Army Institute of Research in Kisumu, Kenya, and his residency and fellowship training at the hospital of the University of Pennsylvania. For nearly 25 years, Dr. Kurtis has studied malaria immunity and vaccine development and has developed several novel malaria vaccine candidates. Two of the potential vaccines have moved through animal trials, and Dr. Kurtis is poised to begin a phase 1 FDA human trial as soon as funding is made available. Dr. Kurtis is principal investigator or co-investigator on 6 current NIH grants, and he's received the Bailey K. Ashford Medal from the American Society of Tropical Medicine and Hygiene for distinguished work in tropical medicine. Earlier this year, Dr. Kurtis was nominated and accepted as a member of the American Society of Clinical Investigation for his work on malaria antigens and vaccines. And most recently, as we'll discuss more later, in April 2020, Dr. Kurtis's research was featured in Nature, one of the top international scientific journals, which further validates his groundbreaking research and its far-reaching potential to save the lives of children. Thank you so much, Dr. Kurtis, for joining us today.
Wow, Robin, that was quite an introduction. I'm glad we didn't rehearse that. I would have been shades of red during it. That was really, really kind of you. I'd like to thank you, Robin, for arranging this and your whole team. There's a whole group of folks behind you that really help put these things together, so I really appreciate that. And lastly, I'd really like to thank the attendees spread out all over the world, mostly for your interest in doing something that the world really needs to address, which is the global pandemic that is malaria. So I think we're going to start with a short film, which highlights a little bit of the work we've been trying to do here at Brown. It features a much younger version of me, and so I'm a little, I get a little sheepish. You'll notice no gray hair, but I think this film will introduce the whole area that we want to talk about today. And then we can jump into some more directed questions, first from you and then maybe take some from the audience.
That sounds great. That sounds great. So let's roll the video.
Malaria is the single greatest killer of children on the planet. We're talking about one child every 15 seconds. This is a fundamental force of nature on this planet. Where malaria's prospered, people haven't. They haven't gone through industrial revolutions. They haven't had political advances. They haven't had the opportunities for advanced education. All these things are due to a sick populace. Malaria is a tiny little single-cell parasite. You get it when a female mosquito bites you. You have tremendous fever, cognitive impairment, and a whole slew of other morbidities. People have been working on vaccines for malaria for almost 100 years. Here at Brown, we take a unique approach. We go to these very malarious areas. Each child has more than one infectious mosquito bite per day. Some children get lots of malaria, and some children don't, despite enormous exposure to infected mosquitoes. And here, we try to figure out what antibodies have resistant children made that susceptible children have not made. We've identified new vaccine candidates that have never been described before. It's considered to be the most exciting new vaccine candidate for malaria that's out there. It takes interdisciplinary research to address a problem like malaria. Our team happens to be epidemiology, biostatistics, cell biology, molecular biology, and immunology. That's our team. What that's allowed us to do, start in the field, come back to the lab, go back to the field. And that is a Brown unique perspective. These students carry the burden of the day-to-day operation of this laboratory. They do tremendous work. The real value of these students is not in what they're doing in these lab benches, it's what they're going to do in 5 years and 10 years when they're inspired by this work. We've been ready to test this vaccine in humans for about the last 18 months. It's very difficult to get NIH to pay for that. If we get through a phase 1 trial with a signal of efficacy, pharma will be very interested. Populations are going to be in conflict as long as they are desperate, and they're going to be desperate as long as they're impoverished, and they're going to be impoverished as long as there's malaria. It is our mandate here at Brown and the Alpert Medical School to carry these vaccine candidates forward. To finally address this culling machine in a significant way would be amongst the greatest scientific achievements of our time.
I have seen that video many times, Jake, and it never ceases to move me. Thank you. Thank you for sharing it with us. You and others across Brown University have answered the call to action to lend your support to the fight against COVID-19. Before we dive into the conversation about malaria, given the pandemic that all of us are facing, could you start by telling us a bit about your involvement on this front?
Yeah, sure. No worries. So it's been an unprecedented infectious disease sweeping around the planet, no question about it. And I think if you look at the Brown contribution to addressing this challenge, first and foremost, you've got to recall that our medical school has roughly 1,300 physicians and physician scientists working in our affiliated hospitals. Those folks are frontline, and I mean real frontline. The first couple of weeks, as we were getting swamped without enough protective gear, you could walk down the halls of Rhode Island Hospital and see physicians intubating COVID patients wearing like their mother's foulard as protection. It was crazy talk. So they were really putting themselves in harm's way in order to do what I think of as God's work. And so the pinnacle, I believe, was direct patient care. But there are other layers to it. There was a huge logistics problem. And I think this is a great thing to bring up on a webinar like this. There was a huge logistics problem for testing reagents and personal protective gear. And do you know who came up with the means to deal with this? The alumni of Brown. They actually donated not just cash money, which is always great, but they also, several of them had connections in the manufacturing space for personal protective gear. And we got truckloads of N95 masks, the special masks, truckloads of personal protective gowns and face shields. So that was all delivered to our area hospitals. That was tremendously important. Another aspect, research. So our researchers, many of them have pivoted their own basic and translational science research programs over to issues of COVID. What are good therapeutic agents? What are good vaccine targets? They've been working on those aspects as well. And then lastly, tapping into the academic and clinical expertise of our faculty. So many of us have been called in to, serve or chair state committees and task forces. One of the reasons our state, Rhode Island, is doing so well, is we have enlightened leadership. So we have a great governor who is science focused. And she herself is not a scientist. I don't mean to suggest that, but she understands the importance of a scientific approach to this problem. She reaches out to individuals with expertise. And what boggles my mind, she totally listens to that expertise. And so you see that in our numbers. The state is doing fabulously. This is a disease that can be controlled if it is approached with a disciplined scientific lens. And so I think because the state is so small and the state government and the universities are so integrated, you know, give you an example. Our director of the Department of Health is a Brown alumnus who did her graduate medical training here at Brown, knows all of the infectious disease Brown faculty. So it really just weds the system together, and it allows us to fight with a unified front. Sorry. Great. How's that for sort of a praise?
It's, you know, I think we all are so appreciative here, certainly in Providence, Rhode Island, and around the world for the commitment of the Brown community and the unique approach that we're able to take because of our position in the state and the interdisciplinary approach that we take to all things. So your lab was open during the quarantine, and we understand that you were working on an aspect of vaccine development for COVID-19. But how did that impact your malaria work, and how have you now gotten back on track? And perhaps we could use this as an opportunity to talk a bit more about what led to your discovery of this concept to fight malaria.
Yeah, so it definitely, and I don't want to sound like the complainy guy, it has impacted everybody's life, no question about it. But it temporarily shut down all the Brown labs, including mine. But then something really unfortunate happened. So you know when you go to get one of these COVID tests, they stick the thing up your nose and they put it in a liquid, right? That liquid is called viral transport media, and there was a global shortage of it. And it turns out my lab was able to make it. So we were for about 2 weeks supplying the state and the perimeter areas with viral transport media. It allowed me to make the argument. I actually had to talk to the provost and the president, said, you know, listen, we could open up and make this. And so they said, okay, we'll do that. And, you know, there was a huge, you know, you got to worry about liability and all that. So we were able to make the viral transport media. Once we were back in the lab safely, we were then able to make the argument others can return. So we were sort of paving the way to reopen labs at Brown in a safe way. So social distancing, masks, but it allowed us to get back to what is our primary mission, malaria. Let's not forget. So in this pandemic, we are now all familiar with the denominator 10,000. You know where you were when your country had its 10,000th case, and sadly and unfortunately, you knew where you were when each country had its 10,000th death. Let me just put that in perspective with malaria as we pivot to malaria. Malaria kills 10,000 children under the age of 5 every week. Imagine if COVID, I mean, the one blessing with COVID is that it doesn't typically kill children. Yes, you'll all have heard cases where that happens and it's horrific, but typically it is not culling our under 5-year-olds. Like malaria has been doing, malaria was doing it last year, the year before that, the century before that, the millennia before that. It has been killing 500,000 children, somewhere in some years as many as 2.5 million children under the age of 5 every single year. And so I've been using COVID as a lens to help people gain perspective on how important malaria is. It sounds kind of like it sounds like I'm trying to rank order the infinite miseries, but I think, you know, COVID has opened up our eyes to how vulnerable we are as a species. You know, if we don't focus on these issues, bad things happen. So that's the sort of way I've been trying to integrate it.
Thank you. No, we really appreciate that that perspective. So Jake, going back to your time, your first forays into Africa, could you tell us a bit? You know, I mentioned in the introduction that you were afflicted with malaria, and that, you know, you recovered, thankfully, for us all. Could you tell us a little bit about what led to your interest in malaria and eventually to the work that you've now dedicated several decades to?
Sure. No, it sounds totally hokey, but the truth of the matter is I was your prototypical Brown undergraduate student, completely lost. And I didn't have a focus in my life. I frankly was not applying myself academically as I should have been, and as I try and tell my children to do now. I had a friend who was really a remarkable person. I'm sure you've heard of her on NPR or Voice of America, Monica Brady, a reporter for VOA, who had this amazing experience in Kenya. And I thought, wow, that sounds interesting. I'll try my junior semester abroad, going to the same program. And so I went to that program, and it was such an eye-opener for me. I got my first taste of doing real research. I was doing a population ecology project on the coast of Kenya. But I think more importantly, I actually got cerebral malaria. And luckily survived. You know, cerebral malaria has a 30% case fatality rate. And even in those who survive, a significant percentage of them have long-term neurologic sequelae, and sometimes, like me, psychiatric sequelae. But it truly, Robin, opened my mind. What society can tolerate losing up to 25% of their children? Where I work in Western Kenya, the under 5 mortality rate approaches 25%. It's insane. What society can tolerate that level of disease and ill health and still thrive? And so in my mind, and it's because I'm not a politician, I don't understand economics, of the 3 pillars of things that can either support a society or frankly destroy and retard it, health is one of the 3. And that's where I really focus my energies.
And so tell us a bit about how you focused in and began to identify these children who weren't getting malaria in these incredibly malarial areas in which you were working in Africa.
Sure. So the ecology project that I did as an undergraduate is sort of a lens into understanding my worldview, which is from the level of a population of organisms, in this case, humans, for malaria. So for 40 years, people have been trying to make a vaccine for malaria, even longer, 50 years. They typically start with mice models of malaria in a laboratory that looks kind of like what my lab looks like right outside. Many of these malaria researchers have actually never been to an endemic country, and many of them have never actually seen with their own eyes a patient suffering from malaria. They're very, I call it the mouse-keteer approach to malaria vaccines. If you start with a mouse model, you are very likely to make a malaria vaccine that works beautifully in mice. The generalizability, sometimes for some diseases, the mouse is a great model. They generalize to humans beautifully. Malaria is not one of those diseases. So the vaccines that come out of the mouse models, they go to humans and they typically don't do so well. So we took a totally different approach. We took an epidemiologic approach. If you walk into a holoendemic, so lots of transmission, if you walk into a holoendemic village, by definition, half the children in that village are currently parasitemic with malaria. If you walk into that village, lots of kids have malaria. There's a few kids who don't. And those kids who don't are special. What is special about them? What are they seeing immunologically on the parasite that the other susceptible children are not seeing? That question really, if you walk into these villages, stay and spend some time and talk to people, it hits you in the face that there are kids who acquire resistance before others. And we developed an approach back in, it was like 2008. We developed an approach to identify the parasite genes that encode parasite proteins that are recognized by antibodies that those few resistant kids make. And those genes are not recognized by antibodies that the susceptible kids make. And that thing, we call it a whole proteome differential screen. Sounds like fancy. It's basically just some molecular biology gymnastics. Using that approach, we're deciphering what makes the resistant kid resistant, and we're now on the cusp of transferring that information to the susceptible kid. Let's make that susceptible kid look like the resistant kid. If we could do that, Robin, you would cut the under 5 mortality by more than half. And that was so exciting back in 2008 when we developed that approach. And when I say we, it's a huge team of Tanzanian scientists, Kenyan scientists, American scientists. When we developed that, it was considered so exciting that we got a science paper out of it. And in fact, it's funny, I still keep copies of it right here on my desk just to remind me to get up in the morning and get to work. So that was our first vaccine candidate. We have now applied that same approach of differentially identifying who's resistant, who's susceptible, what genes in the parasite are they making antibodies to. And we've discovered a second vaccine candidate that, as you so kindly pointed out, came out in Nature. I don't have a copy of that paper yet. Came out in Nature a few weeks ago. Between and that vaccine candidate, we've taken all the way through non-human primate studies. So we can actually protect monkeys that are given real life human malaria, malaria that would kill any one of us on this Zoom call. If you give them to monkeys that have been vaccinated with our particular vaccine, those monkeys don't get sick. They clear their parasitemia in about a week. So we are really excited to move that forward.
So that segues perfectly into my next question, which is, what's the process? How does this work? Bringing a vaccine through clinical trials and eventually to market. What are the hurdles that you now are trying to overcome?
Honestly, they seem like Herculean hurdles. It's a Sisyphean task. Oh boy, I'm going Greek, and I'm half Greek, so I got to go there. Discovering the proper targets, I used to think, was 90% of the game. Having now discovered what we think are the proper targets, I realize it might be 50% of the challenge. So the way funding for research works in the United States is lots of focus on the discovery phase, and then this absence of focus on the translation to market or commercialization phase. There's this gap before big pharma will come and pick it up. And so that gap we call the valley of death, because it's where good ideas go to die. And so the real challenge is de-risking these vaccine candidates from the perspective of pharma. And what I mean by that is, let's just pick on Pfizer for a second. I hope there are no alumni that work at Pfizer on this call, but let's pick on Pfizer for a second. They've got good news. Their COVID vaccine looks really promising. Pfizer knows that every malaria vaccine that has been tried for the past 45 years has failed. So there's a huge barrier, right? Pfizer also knows that there are challenges in monetizing a malaria vaccine, and we can talk about that later. It is possible, Robin, to do it in a very financially sound way, but it's a challenge. It's not as simple as, a heart medicine that you take every single day, or a blood pressure medicine that you take every single day, or a male pattern baldness medicine that you would take every single day. You get one shot, maybe once every 5 years. So it's hard to monitor, and the populations that need it desperately are impoverished. So how do you make that work? So Pfizer really wants to see something coming along that they know, they have great confidence is going to make it to market. They don't want to take a chance. And so that's the challenge we're in. How do we get this through a successful phase 1 study to show that it's both safe and have some signal for surrogate efficacy? That's our challenge. I think if we get through that phase 1 successfully with a signal of efficacy, we'll be in a much better place to move it forward. So that's sort of the challenge that we have.
So Jake, you and I have spent a lot of time together thinking about the financial aspects of funding your vaccine candidates for malaria. And as many of you know on the call, my day job is fundraising for Brown. I only moonlight as a co-anchor on our webinars. So Jake, what is this going to cost, and why haven't people come forward? And one of the questions that I'm asked repeatedly is, why isn't an organization like the Gates Foundation funding this?
Yeah, so let me take that last part first. So the Gates did fund us. If you actually look at it's so funny, I happen to still, I'm hoping one day that my mom and dad are gonna read this. So if you look at our science paper, they funded the epidemiologic study. So way back when Bill first got into malaria, he began with what were called the grand challenges in global health. And they were truly grand in that they were $20 million grants, roughly 15 to 20. And we were lucky with Patrick Duffy and I, it's really Patrick's grant, I was a co-investigator, got one of those first grand challenges. And so that set up the cohort of human beings. I simplified picking the resistant and susceptible children just by walking into it. It doesn't really work that way. It was a birth cohort of 1,000 children who were followed every single week with weekly blood films and visits to their house for clinical checkups and care for 4 years. So you can imagine the infrastructure necessary to do something like that. So Bill was in deeply then. The Gates Foundation, in my opinion, and in some other malaria scientists' opinion, has gone almost into a funny silo. Bill wants to eradicate malaria from the face of the earth. And that is a noble goal. but we don't at the moment have that technology. We need a transmission blocking vaccine. So a vaccine that won't protect me, but if I get infected, will prevent me from transmitting to you. That's where he's put almost all of his eggs are in that basket. Our vaccine's different. Our vaccine is to prevent me from dying. Or let me really reify it. An old guy like me is not, I'm not that useful. I'm much more worried about a 5-year-old or a 4-year-old or a 3-year-old.
And you've already survived it, so.
Exactly, barely. But no, I'm really worried about the 4-year-olds. And so our vaccine will cut, if the modeling carries forward, our vaccine will cut the under 5 death rate by about 50, 60%. That to me is a huge number of not dead children. That to me is incredibly compelling. Working on Bill, getting his team. So, we have monthly conversations with the Gates Foundation to try and get them to pivot their strategy to be a little more room under their tent. Right now, their tent is transmission blocking. So that's sort of been where we are in terms of Gates. Our primary funder at the moment, as you mentioned, is NIH, but they only take you so far. The next studies, frankly, Robin, are boring. 2 groups of humans, 15 or 30 or 50 in each group. One gets the vaccine, one doesn't. Take their blood and do experiments in the lab to see, did you generate the antibodies that will kill parasites? So scientifically, if you're reading a grant, an NIH grant, not the coolest science, not nearly as slick as the discovery science is, but it's super important to get this out. If we don't move this forward, it will die on the vine sitting here at 55 Claverick Street in Providence, Rhode Island, in my laboratory, and it will have helped nobody. And so
we're not going to let that happen.
Exactly.
So then down to brass tacks,
yep.
How much do you need? And this isn't a fundraising call, but I think it's useful for us all to know it's a big number, but it's not an insurmountable number. So tell us a little bit about that. And then we move on to talk about what others are doing in comparable areas. Yeah,
sure. So a phase 1 study, before I would do a phase 1 study, I would want to run several additional non-human primate studies. And there are a couple of reasons for that. We have what we think are the right antigens, the right targets to make the vaccine. The formulation, how you formulate them, we have only tried 2 formulations. And so you want to try about 10 formulations first, because you want to take your most effective vaccine into humans. You're only going to get 1 or 2, excuse me, you're only going to get 1 or 2 shots in humans. So you want to have really explored the space in non-human primates first. So it would take about $2 to $3 million to do the non-human primate studies. And then you've got to file for an FDA IND, investigational new drug. That is a bureaucratic process that is, coming back to the Greeks, truly Sisyphean. It is really, it requires a specialist, typically you would contract it out to people who have done it before. So you got $1 million there. You need to actually manufacture the product in a way that is acceptable for human use. It's called GMP manufacturing. That's $1.5 million. And then a couple of million to do the study. So you're looking at $15 million, 10 to 15, to bring this all the way through a phase
1. But based on some of what you've shared with us today and discussions that we've had with others in the past, this is much more efficacious than anything that is being done right now, from mosquito nets to any other vaccine candidate.
Yep.
So before we turn to questions from the audience, can you just give us a sense of the efficacy that this potential cocktail of vaccines would have?
Yep. Yeah, let me, if you don't mind, can I describe how it works?
Please.
And then a little bit about the expected efficacy. So folks have tried to make malaria vaccines that block the parasite from entering the liver. And the problem for those vaccines is that process is over in 5 minutes. When the mosquito bites you, she injects the parasite, it's gone from your blood in 5 minutes. So that vaccine only has 5 minutes to interdict the parasite from entering your liver. Huge problem. Parasite comes out of your liver 2 weeks later and enters your red blood cells. Folks have tried to make vaccines that block that process, The problem is that process is over in 140 microseconds. It's boom. And so very hard target to shoot as it's moving that fast. Our two vaccines are further down in the red cell cycle. We actually kill parasites. Our antibody binds to the surface of the infected red cell, kills parasites. That's the Nature paper. That's the GARP target. We have 24 hours for that to work. Our other target, our Science paper, blocks the parasite from escaping from the red cell. So we trap it inside a burning house kind of image. That target has 8 to 12 hours. So time is really on our side. When we first took anti-GARP antibodies and added them to parasites in culture, there was a fight in the lab. Not a fight but a conversation, a heated conversation. Because when we looked down the microscope at those parasites, they were all dead, every single one of them. And so we thought to ourselves, that's not possible. We must have done something wrong with the culture. It must be contaminated. That experiment has been repeated 50, 60 times by now. The anti-GARP antibodies force the parasite to commit suicide. It's an unheard of mechanism. So the parasite is complicit in its own demise. And which I think is, poetically, coming out of Brown, I love the poetry of that. So we are really excited about our potential efficacy being 50, 60, 70% reduction in severe disease. That is where I think we're going to be. And if we can add a third candidate that we're working on right now, maybe even higher. But I really would like to move forward with the first two.
And so, and remind us of what the efficacy is of everything else that people are doing right now, including treatment once you've gotten malaria, how efficacious is that?
So there's one vaccine that's moving forward. It's called the RTSS vaccine. Mm-hmm. And in its phase 3 study was only slightly less effective than a bed net. So bed nets provide in a whole endemic area, about 17% reduction in severe malaria and death. And the RTSS vaccine is about 16%. So we've got a vaccine that cost over $1 billion to bring forward that's less effective than a 30-cent bed net. And it is being trialed now in 3 countries in Africa. And it's fascinating. After a phase 3 trial, normally it's licensed and out the door it goes, general population. Not this vaccine, because it has a safety signal. So there are 2 safety elements to this vaccine. And there's great controversy right now because it's being brought out in 3 countries with no informed consent. The safety signal is there's a 10-fold increased risk of bacterial meningitis, which no one really understands. It could be a total statistical fluke, but it was there in the data. And the one that really is a real noodle scratcher is the mortality rate for girls who get this vaccine is 2 times higher than for girls who do not get this vaccine. Mm-hmm. So there's, we need to really, before you deliver a not particularly effective vaccine that has this kind of a safety signal, you've got to really understand it. So our competition is weak, Robin. It's really weak. Right. Treatment, let me turn to treatment. So in Southeast Asia, there are places where the parasites have acquired resistance to all of our drugs. Most recently, artemisinin. And that has been detected in sub-Saharan Africa. The history of drug-resistant malaria is it almost always begins near the Thai-Burma border. And within 5 to 7 years is in sub-Saharan Africa. Once it hits sub-Saharan Africa, flood down from north to south. And the advantages and benefits, which we have enjoyed, I've only been the gloom and doom guy, but it turns out the death rate from malaria in 2000 was 2.5 million. And it has dropped down to 500,000 over the last 15 to 20 years, largely due to the introduction of a highly effective anti-malarial therapy, artemisinin. Now, today, we have artemisinin resistance in Southeast Asia, and it's been detected in sub-Saharan Africa. I'm predicting, barring any other event, new drugs, new vaccines, you're going to see our under 5 mortality rate come back up to at least 1 million children a year. So we really are behind in this game.
And many of those treatments are very expensive. And we know that from being Westerners, going to malarial environments. So I'm now going to pivot to some of the questions that we've received from the audience ahead of the presentation today. And I encourage all of you to use the chat function to submit your questions. We're now going to spend about 15 or 20 minutes addressing questions from the audience, so please feel free to chime in. So following on, one of the questions follows on nicely from our most recent conversation about affordability. Will this vaccine be affordable for the people that need it most, and who will pay for it, and how will that all work?
Yes, great question. So a couple of ways to unpack that. The first is, how do we deliver vaccines today to the developing world? And let me just tell you, it's remarkable to me that children in Western Kenya, where I work, have a higher vaccination rate than children in parts of Providence, Rhode Island. It's crazy, and there are many factors that influence that. One is the totally crummy American healthcare system, but the real issue is the incredible vaccination rates we see around the world through the WHO program in expanded immunizations. It's called the EPI program. It began in the 70s, and it began just trying to deliver DPT, diphtheria, tetanus, pertussis. The DPT vaccination rate is over 85% on this planet. And it's largely, it's microspatial, so there'll be areas where there's really very low coverage. But the incredibly high average coverage is really due to the WHO's program. Most, even low-income countries, have added to that several interesting vaccines. Hepatitis B, which is an expensive vaccine, with their powered purchasing, they now pay $5 a dose for that. And at $5 a dose, WHO can afford to deliver that to hundreds of millions of children. So that vaccine, the hepatitis B vaccine, is a complicated vaccine, forming this large particle. It's not an easy one to make. If that can be done at $5 a dose, and remember, it's differential pricing. When you and I got that vaccine, we did not, $5 was not what the amount that was paid for that vaccine. So WHO negotiates differential pricing. There is a program, it's called Gavi, and I can't remember what it stands for, but it is a program / mandate that if you take grant money from several sources, your vaccine must fulfill the Gavi goals, one of which is this differential pricing. So if we can make it at $5 a dose, I know it can be delivered. The strategies, and remember I said something about formulations. One of the formulations that I really want to test in non-human primates, it's called a phage display formulation. Robin, I could make 10,000 doses in a flask this big using that formulation approach, literally pennies a dose. And so we are definitely sensitive to the need of delivering this at pennies a dose. Now, pennies a dose in Lagos, hundreds of dollars a dose in Greenwich, Connecticut. That's the plan.
Right. Thank you for that. I have a few more questions that we'll go through that were submitted in advance, but I made a mistake in alerting people to use the chat function. Please enter your questions through the Q&A function, not the chat function. You can tell that I'm still finding my way along in Zoom technology. Apologies for that. So the next question is slightly more technical, so I'm going to read it directly as it was submitted. If antibodies that stimulate GARP are causing an inhibition in malarial replication, what happens if host antibodies start inhibiting GARP? Is there a possibility of this process stimulating Plasmodium replication rather than shutting it down? And it might be useful for those of us that don't have a medical background for you to clarify the question so that we can better understand it before answering it.
Absolutely. At the core of that question is, hey, Jake, how do you know you're not going to make things worse? That's the core of the question. And so the question is, breaking it down is, your antibody binds to GARP and the parasite dies. It actually commits suicide. How do you know GARP is only involved in activating death? What if GARP is like a switch, forward, neutral, reverse? Forward, grow, neutral, don't do anything, reverse, die. How do I know GARP doesn't do all three of those things? It turns out GARP only activates cell death. And you might ask me, Jake, why does the parasite want to kill itself? Why does GARP exist as a target? And Here's the answer. Perversely, the parasite does not want to kill you, Robin. It wants to multiply inside you as much as humanly possible or parasitically possible and not kill you. Because if it kills you, you're a dead-end host. All the parasites die if you die. So the smartest parasite, and I'm sort of going down, this is a Darwinian process. The smartest parasite, the best adapted parasite, senses its host's health. If the host is looking unhealthy, it shuts replication down. That would be the smartest parasite. I'm a strong believer. We actually hinted at it in our Nature paper. I think GARP is on the surface of the infected red cell to sense host health. And when the host is unhealthy, it shuts down parasite growth. We are activating GARP with an antibody. That's the vaccine approach. Antibody binds to GARP, shuts parasite growth. In fact, parasites kill themselves. So that's, I think, the answer to the question. We have never seen enhanced growth with anti-GARP antibodies. And I personally don't believe it's possible.
Right. Great. So I'm going to turn now to some of the live questions coming through from our audience.
Awesome.
There were several questions about students and PhD candidates in your lab, and people are curious about how you include the broader Brown community in your work.
Yeah, absolutely. So, this takes a huge team. 20 or 30 scientists and healthcare workers and village health assistants in Kenya on the payroll right now, Tanzania as well. So huge endemic country participation. Multiple labs. If you look at our Nature paper, it had 33 authors from 15 institutions. Huge collaborative projects. In my own lab, we're a lab of about 20 folks. We tend to be postdoc and junior faculty heavy, but we also take undergraduates every year. So I always have undergraduates paired with my postdocs. And for a variety of reasons. One, you got to get your start somewhere. And if we're not successful, I want to have been involved in training the next generation to carry the ball down the field. So we always have undergraduates in the lab, and we currently have 2 grad students in the lab, one working on malaria and one working on another tropical infectious disease I haven't even mentioned called schistosomiasis. So we love having the graduate students in the lab.
That's great. And I've noted from visiting your lab, it's really fun to see the different ages and faces and the diversity.
It's not just gray hair.
People that attract what you do. And it's people who have experienced malaria themselves firsthand, but also people who want to be a part of this, extraordinary discovery that you're on the verge of finding.
One of the great things for the students, the Brown students in the lab, is we routinely have our Kenyan and Filipino scientists come to the lab either to train us in something they know how to do or for us to train them in something we know how to do. So there's constant interaction. I think to become a global health physician or physician scientist and not touch and interact with the people that are suffering from these diseases is a great mistake, because at the end of the day, doing lab work isn't that different than accounting. It's the thing itself. What motivates you is the impact you can have. And to not see that is a travesty. So the grad students are constantly interacting with our folks from overseas. And if it weren't for COVID, one of my grad students would have been in the Philippines with me this past March, and the other grad student would be in Kenya with me over the summer. Unfortunately, neither one of those experiences is going to happen this year.
Well, it will soon enough.
Exactly.
You mentioned another research project that you're working on, and some others are curious what else is going on in your lab. And once you've successfully launched this vaccine and are saving 5,000 to 6,000 children's lives a week, what will you do next?
So let me take the first one. What else is going on in the lab? So in the broader group, the Center for International Health Research, it's actually led by a woman named Jennifer Friedman. She's an MD, PhD, MPH, pediatrician, epidemiologist. She's running a clinical trial right now in Uganda And it should have also been in the Philippines, but there was some truly political challenges with a dengue vaccine that shut down studies in children in the Philippines for a very long time. But she's looking at new ways to treat children at younger ages for schistosomiasis, because right now we don't treat children under the age of 4. They continue to have schisto, and they accrue the morbidity of having a worm living inside your liver, basically. So she's running a randomized controlled trial of treating children under the age of 4. She had just completed a randomized controlled trial of treating pregnant women with schisto, because pregnant women weren't treated. And we know any inflammatory process in your body is bad for pregnancy. And having worms in your liver is like number 1, number 2, and number 3 for having inflammation. So, so there's a whole drug treatment arm to what we do. There are vaccine approaches for schistosomiasis that are copy-paste the malaria approach, the same thing happening in schisto. We have a PhD scientist who is working on the immunology of the placenta as it relates to dominantly schisto, but a little bit in malaria, because we know that when mom has an infectious agent, doesn't matter what it is, it's not great for pregnancy. And if you're in a low-income country, any insult to your pregnancy is a serious threat to the health and well-being, not only of the mom, but also of the newborn. So the super important aspects there. I told you antibodies to GARP kill the parasite. There are tools, they're called monoclonal antibodies, where I can make that antibody in huge buckets for use as a therapeutic. And let me put this in perspective. Hopefully none of you on this call have had to deal with cancer in your life or with one of your loved ones. But if you have, the new therapies for many cancers are antibody-based. The revolution in treatment of breast cancer, Herceptin, is an antibody, a monoclonal antibody that binds to and kills breast cancer cells, certain kinds. We have a monoclonal antibody that binds to GARP. How would you use such a thing? There are parts of the world, let me give you an example, northern Mali, where malaria is seasonal, lasts for about 3 months. What if we gave a shot of antibody at the beginning of the 3-month period? They would be protected through the 3 months. So seasonal malaria prophylaxis would work beautifully with this monoclonal, and the travel market. And in my mind, I use the travel market to pay for the vaccine. So there's, you gotta, it's real politics. That Pfizer needs to make its pound of flesh. Let it make that pound of flesh on a population that can afford it, whilst providing a service to a population that can't afford it. We're also currently working on antimalarial drugs. So small molecules that mimic the binding of the antibody to GARP. If they mimic that, they're going to have the same effect. So those are sorts of the projects that are going on right now.
Right, that's great. And as we zoom out a bit further, you have leadership responsibility for MD, PhD training and a number of other areas within the Warren Alpert Medical School at Brown. Can you give us a sense of why, in our time at Brown in the 1990s, medical school was always known for being very good. We were working on some interesting research projects, but in the past 25 years, it really seems like there's been a huge investment in the research that Brown is doing in biomed. Could you give us a sense of what's going on?
It has absolutely exploded. So we right now, things happen when areas coalesce. And what we have coalescing right now is the holy trinity of leadership. We have, actually it's 4, so maybe it's the 4 horsemen, but that's bad, that's the apocalypse. We have 4 individuals involved right now. We have a chancellor, Sam Menkoff, who absolutely recognizes the power of the medical school. We have a president and a provost who are fully invested. They look at Brown. Their job is to take Brown to that next level. And ask yourself, is the next level hiring another professor of English studies? Does that transform Brown? Or does hiring a brain sciences institute transform Brown? And you can make both arguments. I'm trying to, I obviously have my own opinion, but they have voted with their feet. They believe that the medical school was pretty good, needed some buffing and some polishing, and that's their diamond in the rough. So provost, president, absolutely on board. And last but not least, We have a dean of biology and medicine who is full on awesome. First off, he himself is not just a great administrator, he's a serious scientist. He's in the National Academy of Medicine. His list of accolades is insane. He gets what a great medical school can be and what it can do for its university. And I think with all four of those individuals in voting with money and resources, our medical school has risen up. I think we are the powerhouse waiting to be unleashed at the university. I really feel that.
Well, we certainly feel it here on campus. So a question, a very specific question. What type of reading would you recommend for people to become more knowledgeable about malaria?
Do you know the first thing I have my students read? It sounds totally crazy. The wiki page on malaria is really pretty good. So I would start with the wiki page just to get your background done. There are some fabulous books about the impact of malaria. So, even a bigger topic, the impact of infectious disease on human civilization. So Jared Diamond has a great book, Guns, Germs, and Steel. And it's about the three things that Westerners brought, or Europeans brought, to the developing world and how they decimated populations. And so I think it's a great perspective. And malaria, of course, has a chapter or two in there. That to me would be a great initial read. And then if you want to go even deeper, there's a book called Malaria Capers. And it's about mismanagement of funds in the early phases of malaria vaccine work, which I think is 40 years ago. So you won't read about me in there at least. But I think those three books would be a great place to start.
Great, thank you. I think we probably have time for maybe one or two more questions, depending on the length of our conversation. I'd like to ask you a question about one of your most recent awards that you won from the Brown Biomedical Innovations to Impact Fund. Congratulations.
Thank you.
And my introduction to you was long enough. I didn't think I could squeeze it in then, but I thought if we had time, I'd like for you to tell the audience a bit about what BBII is. Yep. And what this fund will mean in terms of advancing your work in malaria.
So BBII is an initiative that is predicated on the notion that Brown historically has not participated in the bench to bedside transition. We've been very bench oriented. Our ideas have stayed in our laboratories. And in order to get to the bedside, you have to de-risk. You have to do experiments that are not necessarily scientifically the coolest, but are practical. How do you discover a new antibiotic in your lab? The skills it takes to do that are sometimes quite different than the skills it takes to do the preclinical studies and show that it's safe and effective in humans. And so Brown hasn't really engaged in that process sufficiently. The BBII fund is designed to, first and foremost, bring venture capital and to some degree even large foundation money into Brown to encourage Brown scientists to begin to encourage and facilitate them to do the kinds of experiments that allow their innovations, their discoveries to be translated into a marketable product. That's the vision for that whole group.
And that fund is still open. I know we've received a number of large grants and gifts to it over the past couple of years, but it's still open, right?
Yep, it is. I believe, don't hold me to this, it's between $8 and $10 million that's in. They are giving out 4 or 5 $100,000 to $150,000 a year awards. And there is no question, when I'm looking out at the research buildings right now, there are grapes on those vines that will wither without the translational support. So it's super important. And again, our dean gets it. Our dean understands that this is a priority. And the great thing about it, it's a priority that will pay dividends. So when Brown licenses out a product, there are royalty streams that come back. So it really is, and in fact, BBII is specifically set up to be evergreen with royalty licensing payments. So it's a great way to invest in Brown.
now please tell us about the grant that you won from the BBII.
Yes, so that grant is to commercialize the monoclonal antibody against GARP. So if you have the GARP protein on the surface of the red cell, the antibody binds, sends a signal, parasite dies. What if we could make this antibody in huge quantities? It needs to be a monoclonal antibody. So there's engineering involved, but not physics. So we know antibody binding, dead parasite. All we got to do is make lots of this. So we know the target. The grant is designed to, this is a funny mouse versus human story. So we tried making the monoclonal antibodies in mice. They work. When we try to turn that mouse, you can't give a mouse antibody to a human, you have to humanize it. When we did that, it falls apart. So we're like, hang on a minute, why are we starting in mice? So this grant is really about starting with human beings that have very high antibody levels to GARP, taking their B cells, which are immune cells in their blood, which make antibody, taking their B cells and screening them one by one for antibody to GARP. The B cells that make good antibody to GARP will be sequenced. We'll know the gene for that antibody. We'll clone it. Once we've cloned it, we can make buckets of human anti-GARP monoclonal antibody. Three places we can use it. Let's start with the money first. Travelers. You want to go for 3 months to Kenya, get a shot of this antibody, off you go. Seasonal malaria immunoprophylaxis, the northern Mali kind. So about half of the people who live in malarious areas live in seasonal malaria areas. And so it could be very useful there. And the third is if you have severe disease. Remember I told you the GARP target lasts for about 24 hours, and it's the form of the parasite actually that's not circulating in their blood. It's the form of the parasite that's stuck in their tissues. It's one of the hardest forms to kill with a drug. Our antibody kills that. And so the idea would be to use it for treatment of severe malaria. So our GARP vaccine maybe cuts severe malaria deaths in half. What about a GARP therapeutic monoclonal to mop up some of the rest? So there's 2 ways to go about that. And that's what the grant is about, identifying the human monoclonal. And then of course, there'll be other layers to get that further advanced, but that would be a great advantage.
Dr. Jake Kurtis, this has been a fantastic hour-long conversation. The questions have been wonderful. I'd like to thank you for your time. I'd like to thank our participants for their joining us today, for their great questions. Please feel free to follow up with me or any of my colleagues. We can facilitate one-on-one conversations with Dr. Kurtis if that is of interest. We look forward to continuing to follow and support your work and wish you the best of success in moving these exciting vaccine candidates forward.
Robin, thank you so very much. And to all your staff, I really, really, really appreciate you taking the time to interview me. And I really want to thank the participants out there on planet Earth. Thank you so much for your interest, and would love to chat with you in the future. Thanks so much.
Great. Thanks, everybody. Bye-bye.